For days 8 homework, there were 5 questions. The first one just asked you to show where Hox 1, 2, and 3 would be in an embryo. Each Hox gene in my hypothetical embryo is in a different segment, so this would be segment 1, 2, and 3, and Hox 1 in these cells, Hox 2 in these, and Hox 3 in these, the 3 segments. Now in reality, Hoxes can overlap, there's more complexity, but this will work conceptually, and in some cases the Hoxes don't overlap, so it fits at least part of the time. This person also showed that this whole set would be dorsal. I just wanted to point out, you don't have to write dorsal over and over. You could write dorsal in the first one and then just D. You could write dorsal in the first one and just do an arrow to show this. So, and instead of Hox 1 and Hox 1 and Hox 1 and Hox 1, you could just write Hox 1, H1, 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 do Hox 1. So you can do things that will save you time, and I'll understand them. The second question asked for the structure of a gene that would be expressed in a specific cell. Here's that specific cell. And what you have to be aware of is, what is the set of transcription factors unique to that cell or to those cells? So in this case, this is the only cell that has Hox 3. So Hox 3 here, but it has Hox 3 and ventral, which is just Hox 3 and ventral is just these two. And then Hox 3 ventral and segment anterior. That's an ANT. So this is the only cell that has those 3 proteins expressed. So all cells have all genes, but only this cell in these 8 has those. So a gene with this structure, here's the gene. It's gene X, meaning it's going to produce messenger RNA for X and protein X. So gene X needs a binding site for Hox 3, for ventral, and for segment anterior. They all need to be enhancers, and they all 3 are required. So this person correctly listed all 3 as enhancers, all 3 required. That's going to get us protein X. The next one was number 3, to show which cell it would be expressed in, and this same image shows it's only going to be this cell. So that was the first 3. Here were a couple others that showed it. Here's our hypotheticals. Notice this person just did H1, H2, and so on. And they used this cell, so H3, segment anterior and ventral. They're only going to be expressed in those cells. And so we're going to get gene X, and they said they're all enhancers. And they say, if not all 3, then no gene expression. So again, correct. Here's another one, just showed a messy embryo, so they're actually skipping some of the cells. It would be like this, and I'm fine with squares instead of circles if you want to do it that way. And it's this one, again, Hox 3, ventral, segment anterior. That would be this one. They're all enhancers, we need all 3. And here is the protein. They didn't say it's protein X, but I knew from context the question asks about protein X. So in this case, even though it's not labeled, I would have known. If it's ambiguous, though, you definitely want to label, and when in doubt, definitely label. Another one, here we go. There's Hox 3. Zooming in, you're going to have Hox 3, ventral, anterior segment, need them all. They're all enhancers, they're all necessary for gene X to be expressed. Again, gene X wasn't labeled, but in this context, I know this has to be gene X, or this whole thing is gene X. The next question was about metamerism and cephalization. This person drew 3 segments. They decided Hox 1 would be anterior in this segment, Hox 2, Hox 3, Hox 4, Hox 5, Hox 6. That can be, more often you'd expect Hox gene to be expressed all the way through a segment and maybe even into neighboring segments. But hypothetically, this could happen. And so cephalization, here are claws that only appear in the front. Here's eyes that occur here, but not at these other sites. There's a heart that occurs here, but not here and not here. They've been X'd out. And then, so that is metamerism, repeated structures in different segments with some suppressed in some segments. And then cephalization, the head end has some additional features. They drew the gene for Tin Man. I'd actually asked for PAX6, but conceptually they're the same. So they decided the heart is going to be here. Here's their heart. So that means HOX2, dorsal, and segment anterior. So it doesn't quite match their image because HOX2 would be here. But if this was where they wanted the heart, then the gene structure should require HOX2, dorsal, and segment anterior, and that would get you Tin Man messenger RNA and then Tin Man protein. So on this one, I probably would have taken a little bit off because up here they say HOX4, down here HOX2, and it's not quite clear what the location is that they really want. Oh, wait a minute, actually this green line puts the heart up here. So I think they drew this first and then later decided they wanted the heart up here, and that's what this would do. For PAX6, you would need to also include what is it that makes the eyes be out on this lateral side. So you have to invent a transcription factor for lateral versus one that's expressed here, medial. So lateral would be expressed here, and medial would be expressed in the middle. And there are transcription factors that do that. So here is our developing organism. Here are the eyes, antenna, and Tin Man. Not sure why this says PAX6, because I don't think it needs PAX6. Not sure. So, okay, so where are you going to get the eyes? Well, this person said HOX1, which would be anterior. These HOX2 and 3 are not going to be there, so they're not going to bind. Or if they do bind, they might be repressors. That would block the eyes back here. And a little hard to read, but this one is anterior. I think they meant segment anterior. Posterior, not binding. And I can't tell for sure if they're dorsal or ventral. When it was a little less pixelated, it was clear. But you'd also need a lateral. So one of these should be lateral or edge to show that it's going to be out here and not in here. For PAX6, they showed a different set, and this time it's HOX2 and transcription factors. And then for Tin Man, it is HOX3 because they wanted the heart back here, and they used posterior, should be segment posterior. But the idea is correct on all of these. So I think I took off a tiny fraction of a point, 1% of the whole thing, showing that in different regions, you're going to have different combinations of proteins and binding sites to make that work. Here's another one. I like this one because it was simple, though it's missing one piece, although not as sketched, so I think I gave this one full credit. So they only have 2 segments. They have eyes here, but not here. You can't really see the X very well, but those are X'd out. So the eyes are here, and the heart is here. It is metamerism. It is cephalization, because here we have eyes, here we don't. And they put HOX1, 2, 3, 4. You're going to be better off putting 1 HOX gene per segment, just to keep it simple. But they had, for PAX6, they have HOX1, which is this region. And they have ventral, which would mean it would be toward the ventral side, and they did segment anterior, which would be up here. The thing is missing, I think I may have taken off 0.01 point for this, would be lateral. And I realize that we haven't talked about yet, but part of what I want you to be able to do is infer or invent logical things that fit with what I expect you to know for these kinds of questions. And then back here, Hawks 3, this is more or less the area of Hawks 3. Dorsal, they decided it would be on the dorsal rather than ventral surface. Segment posterior, which would put it back here, except that would actually make it more like Hawks 4. So on an exam, I might take off a little for that inconsistency, but I didn't worry about it here. For the last question, you had to use this image to check on whether this was accurate, and then what's the height here, and what's the side-to-side distance. So a number of people went in the wrong direction. Let me talk about the right direction here. So what we have, you can see here, series of black lines, and those are all membranes. They are going to be your most accurate way to assess, if you can see something that is a membrane, your most accurate way to estimate size. What you needed to do was count not just the black lines, 1, 2, 3, 4, 5, maybe 6, 7, or 8, depends a little on where you count them, but also the white spaces in between. So let's say here we've got 1, 2, 3, 4, 5, 6, 7 lines, and then we have 1, 2, 3, 4, 5, 6 white spaces between them. That's 13. The white spaces are about the same thickness of the line, so 13 times 7 to 8 nanometers gets us to approximately 100 nanometers. If you round it up to say 13 times 10, you'd get 130 nanometers, and I'd say close enough. So even if you round the size, the thickness of a membrane, it's close enough. Remember, these are often a little blurry, so this may be looking a little wider than it really is, but looks like that is either accurate or very close to accurate. Okay, so if that's 100 nanometers, that is about the thickness here. Some people went the thickness this way, in which case, if you showed me this, you'd get closer to 250, 300 nanometers. And then the distance here to here is, ballpark is about 10 of these from here to here. And so that's in the neighborhood of 1,000 nanometers or 1 micron. And here's their logic. Top to bottom, 100 nanometers. Side to side, they went with 700. That's fine. And the membranes are about 10 nanometers. Here's another one. And I can't see that they were using the membranes. That's what it looks like to me. But that is around 100 nanometers. I would have preferred that they say membrane about 10 nanometers and about 10 membrane plus white space. On an exam, do show me your work, show me how you made the estimate. There were some errors here. Some people decided, well, a whole cell is 10 microns, but you can't actually see a whole cell here. This is only a part of a cell. And even then, cell sizes vary tremendously. There are some that are well over 100 microns and others that are quite tiny. Also, cells can be really thin, so sometimes it looks like you have a whole cell, but you really only have a part. And then Golgi, some people just estimated the approximate size of a Golgi, but those also vary tremendously in size, so that's not going to be accurate. Some people decide this was a mitochondrion, but a mitochondrion should have some variation. Oops, let me try that again. Outer membrane, and then outer membrane, and then an inner membrane that has lots of folds. And you can usually see these folds inside. This white space inside would not be accurate. And a mitochondrion really would be closer to this size. I don't even see anything that looks like a mitochondrion here. Definitely not a nucleus. Again, nucleus would not be pale white inside. So over time, I'd expect you to realize if this is a membrane, then this can't be a nucleus, and this can't be a whole cell. There were some answers that as long as their logic was correct, I counted that came up with different numbers. As long as I looked at it and went, yeah, I could see how you could come up with that.